The Array Studio DNAseq pipeline runs Summarize Variant Data, or Mutation to SNP. This identifies variants in your aligned DNAseq or RNA-seq data relative to the specified reference genome. You can also run this module under NGS, Variation, Summarize Variant Data. Specify your input project and data, which is an NGS data object, whether to analyze the entire genome, certain genes, or certain genomic regions, which samples to analyze, specify an output name. You can also change pileup quality options such as base and mapping quality and how to deal with duplicates and multi-mapping reads. You can also change how many total reads and mutant reads should cover a position to be called as a variant. Under the Advanced tab, you will find additional variant filtering options, and you can specify what kinds of reports to output. By default, a merged table object will be created for all samples. You can also output individual variant reports for each sample. Array Studio is in the process of moving toward variant call format specification-based reporting. This allows more efficient streaming of data from files. Annotation columns are more organized, and there's better compatibility with external tools. You can choose to output a merged VCF file or individual VCFs for each sample. You can choose whether or not to annotate by the compact dbSNP database for known polymorphisms and change SNP calling cutoffs. These values are the same used in Illumina's Cassava software. You will want to specify an output folder because the intermediate files generated by this analysis can be useful for downstream analysis. The default output, such as you would get with the pipeline, generates one table, each row is a variant, the frequency of each variant in each sample is reported, and matches to the dbSNP database will be indicated, such as common, or dot, or flagged, where flagged are usually of interest. If you wish, you can right-click on a variant ID and choose to open a genome browser to directly view the coverage and variant data. You can also annotate your variant report under Annotate Variant Table Report. This will use selected variant annotation databases to provide allele frequency information as well as predict functional consequences of each mutation. However, as mentioned before, Array Studio is moving toward VCF-based variant annotation.